Since it becomes more and more important to infer unknown biological functions by integrating multiple type omics data into networks or mapping the data across species, Omics Analyzer provides biologists with an interface to import multiple omics datasets across different data types and species and manage the data easily in Cytoscape. Next, we will show you how to use this. After successfully loading, you will see a network like this. Now, we get one gene expression experiment file to associate with the network. The file describes an experiment on Arabidopsis. Go to the Omics Import panel. First, choose the Name the Mapping ID for Data File option and select the Arabidopsis probe set option. This option allows the attribute mapping information to be labeled with a user-specified ID. We use this option here because the expression data uses probe IDs, while the network uses gene IDs, and we must provide a mapping file between the two types of IDs. The next step is selecting the Input Experiment data file. Click on the Select button and browse the sample data folder to find the experimental data file. Data files can be in the form of a tabbed text file or an Excel spreadsheet, where each row of the file is a set of measurements of a probe set or compound, and each column is a hybridization or condition. Select the mapping file that maps Arabidopsis probe sets to Arabidopsis gene loci. Select the mapping file named mapping atprobes to atloci.txt using the next selection box. The reason we need the mapping file is to allow users to map between probe set names and locus IDs. We can also use it for other things like mapping ortholog genes and biological compounds between different species. This step is necessary because known names in the graph do not always correspond to the names of nodes in the experimental data file. An important note is to always keep the first column of the mapping file as the same as the current network node ID, and the second column as the ID that the node maps to in the data file. Choose the current node attribute to associate with using Choose an Attribute to Assign Values to Node. Set the value to Show for this example. This tells the plugin what field in your graph file you should use to match the names. The drop-down box gives a list of attributes for the current graph being displayed. An important note: to match the node name using data files from the MetNet database, you should choose Show. Other pathway data sources may use different field names, and you should use the proper values. Once the proper fields are filled out, the Import button will be available. Click Import and import your file into your current network. After your import, a result window will pop up specifying the number of nodes that matched those of the current network and the number of columns of data which were read in. Here, it shows the results of a successfully imported data file. We see that there are 36 nodes that were matched from the data file under 13 different conditions or columns. If anything goes wrong, click Cancel to start again. The Reset button will reset all values if you want to import another data file into this network. Click Selected Nodes on the bottom left. To highlight the nodes in the graph, left-click on any row of data in the data panel. The nodes which have experimental data will be listed or highlighted. The matched nodes will be selected and their node names will be listed in the data panel. To highlight the nodes on the graph, simply select any node in the data panel. Additional node information, such as the node name, expression values, or others, can be displayed in the data panel by right-clicking the mouse on the data panel icon and checking the boxes for the properties that you want to see. Click Save pvals file to save the mapped data file. This can save time and allow for comparisons between different mappings. A p-valves file for the current network will be created in a directory you select. This file contains the expression data for the matching nodes in the network. Nodes that did not match any experimental data will not be included.